Hi, class. This is the recorded lecture for Chapter 1, Microbiology. On the first slide, you'll see that we have some study objectives. And what I want you to do with these after the lecture and when you're studying is go back and practice, make sure that you can answer all of these things. So that's a great way to practice and to study for the test. So the early years of microbiology, it's really hard to put an actual start date on when microbiology began. Certainly, once we were able to see microorganisms and understand and recognize that there's a whole world, a whole level of life that we couldn't previously see, then we could say that's the beginning of microbiology. So some people would say it was when Leeuwenhoek began making and using simple microscopes. And he was able to see tiny animals. And some animals are tiny, but you can still see them somewhat with the naked eye. And some animals are too small to be seen with the naked eye. And typically we're talking about parasite types of animals like worms and such. But he was able to see those with the microscopes that he made. And fungus, algae, and protozoans. So these things he could see with his microscope. He called these things animalcules. And you can tell it doesn't roll off the tongue that easily. Luckily, we changed that name to microorganisms. And so microorganism, that term can be used for anything that is too small or virtually too small to be seen clearly with the naked eye. One thing that Leeuwenhoek did not see with his microscope, which are also included in the group of microorganisms, are the bacteria, the archaeans, and the viruses. So his microscopes were not good enough to see those things. But here you see in the top left a painting of Leeuwenhoek looking through his little microscope. On the top right, you see a recreation of what his microscope was like. And you can see a little needle-like structure that hangs kind of above where the lens is, a tiny little lens. And in the bottom center, you see more of a modern day picture of what you can see under a microscope. And one thing to pay attention to when we're looking at micrographs, a micrograph is a picture that's made through a microscope. So micrograph is a combination of the two words microscope and photograph. But one thing you want to notice is the LM stands for light micrograph. That means this photo was made using a light microscope, which is the one we're going to use in lab. And then and the one that typically students have access to. And then 50 micrometers is the scale. And so you can see that a length of about this much is 50 micrometers. And so you can get a sense of how big this object is here. So this paramecium is probably about 250 micrometers. And we use this Greek letter mu to represent micro in the metric system. And then the M stands for meters. So that is about 250 micrometers. So it's a good way, when you have that scale, you have a sense of how magnified the item is. You can estimate the size of it. So you may recall that there was a taxonomist named Carolus Linnaeus who had developed a taxonomic system for naming plants and animals and grouping them. And we've expanded that system to include other organisms, of course. And so we're going to be talking about the classification of different microorganisms. And by keeping track of the classifications, we can keep track of what the characteristics would be for each of these different microorganisms. And it's important to be able to identify microorganisms based on their characteristics, because identification of microorganisms is really the most important part, or the most important first step of helping to treat, to get to a treatment for some kind of infectious disease. And so if somebody has an infectious disease, then identification of the microorganism that is causing the disease is the first step, and that leads you to the correct treatment. So correct identification is really important, and not just correct identification, but correct and fast identification is probably the most important, so that the patient doesn't continue to get sick while you're trying to identify it. So we want to correctly and quickly identify a pathogen that's causing somebody's disease, so that we can then go to the correct treatment right away. So like we said, Leeuwenhoek, his microorganisms actually only included these things. The things he saw were fungus, protozoans, algae, and animals. And then we've added in these two here, and I haven't, we will talk about virus as well. But these are the main groups of microorganisms that we're going to be talking about during the course, during the microbiology course. And so we're going to briefly touch on each of these right now, but then this is an overview really for the whole course. So we will spend several chapters, quite a few chapters on bacteria. We're actually not going to talk very much about archaeans because they do not cause disease, and we are particularly going to focus on pathogens. Pathogens are microorganisms that cause disease. And so because archaeans don't cause any diseases that we've ever identified, then we don't talk about them very much. They're actually very interesting and quite useful in a lot of ways, but this course is going to focus quite a bit on kind of a medical angle for microbiology. So we'll talk about them a little bit. And then the fungus, the protozoans, the algae, and the animals. So bacteria and archaea, you may recall that these are prokaryotes. That means they do not have a nucleus. It also means that they are unicellular, because all prokaryotes that we've ever found are unicellular. Now, prokaryotes are much smaller than eukaryotes. In this picture here, you have two human cheek cells kind of overlapping. And then that's eukaryotic human cheek cells. And what you can see here in a very small little what looks like little dots, these are the bacteria. So in this happens to be Streptococcus, what you would call strep, in the mouth. And so you can see that there's an enormous size difference between the human cells and the bacterial cells. So, and you can see here, this is a light micrograph. So it's a photograph made on a light microscope. And this scale is 20 micrometers. And so you can estimate the size of the human cells is maybe 60 to 80 micrometers across, maybe 100. And the size of the strep is maybe 1 to 2 micrometers. And so there's such a huge differential in size that you really wouldn't make an error. You wouldn't accidentally identify a prokaryote for a eukaryote simply based on the difference in size. So that's an important thing to know. So that's another reason you have to pay attention to the scale measurements that are given to you in photographs. Bacteria and archaea reproduce asexually. They divide by something called binary fission. And binary fission is very similar to mitosis that we know about in the eukaryotic cells. And it's just a way of a cell dividing to make two genetically identical cells. And so because these are unicellular organisms, there's one cell makes the whole organism. That means that when that cell divides, the whole organism is reproduced. So that is the way that the prokaryotes reproduce. All right, moving on to the fungus. The fungus are eukaryotes. They do have a nucleus. They are heterotrophic. That means they're not photosynthetic. They don't do photosynthesis. And they get their nutrition from other organisms. And generally, they are decomposers. They absorb nutrients from organisms that are decomposing, and they actually contribute to that process. They have cell walls. Their particular cell walls are made of a carbohydrate called chitin. and you do pronounce this CH as a hard CH, chitin. And the fungus, the group includes two main categories, the molds and the yeasts. And both of them can be helpful, and some of them can be pathogens. And of course, we'll focus a lot on the pathogens. But there are some molds that are pathogens and some yeasts that are pathogens. So on the picture here on this screen, you see on the left, you see molds, and on the right, you see yeast. This little symbol here stands for scanning electron micrograph. So it's a photograph made by using a scanning electron microscope, and we'll talk about that a little bit later. Micrograph. So SEM, and you can see the size here is only that legend gives you a 5 micrometer measurement. So these are small. All right, protozoans. Protozoans are eukaryotes, unicellular, and they are, some of them are similar to animal cells, some are similar to plants, some have chloroplasts, some don't. There's a lot of variation in this group, but they're always single-celled, and they can do asexual and sexual reproduction, and typically they live in water, although they can live in animal hosts, and some of them can be pathogens. We will look at some protozoans in lab, but they won't be alive if they are pathogens, and if we do look at live specimens, they will not be pathogens. But the main thing about protozoans is they have different means of locomotion. That means movement. Some of them have flagella, cilia, and some of them crawl. So this amoeba at the top here, he has this way of moving that involves a flowing of the cytoplasm, and he crawls through the environment. So this one of the interesting things about the protozoans is how they are able to, different ways in which they're able to move. And so we say that they are motile. That's the word we use. It refers to the movement. So protozoans, most of them are motile. Algae are closely related to plants. Some algae are unicellular, some are multicellular. Very few are pathogens. And so that means that we are not going to talk about algae very much. Actually, I don't think we talk about any of the pathogens in this course. It would be so unusual for somebody to have a disease caused by an alga. And so that's, so we won't talk about them too much, but you may recall, like plants, they're photosynthetic. They do reproduce sexually, and they can be identified based on their various characteristics and chemical composition. But the alga are not going to be a focus for us in this course. And of course, we're interested in parasites. Some parasites are, parasite is not a particular group. Some of the protozoans can be parasites. Protozoans. Some of the animals, and particularly worms and arthropods, can be parasites, like a tick. And there are worms that can be parasites. So parasites refers to organisms that tend to cause disease by, living off of another organism. And then, like I mentioned before, viruses. Viruses are very tiny. Viruses are not going to be able to be, we're not able to be seen by Leeuwenhoek, but now with better microscopes, we can see those. And so in this picture here, you have a worm that's a parasite. And over on the right side, you see this is a virus attacking a bacterial cell. And so just like bacteria are much smaller than eukaryotic cells, viruses are much smaller than bacterial cells. So I'm going to take a break, and you can study the material and then move on to the next lecture segment.